Good morning, everyone. Good morning. So, I will proceed to the live. So, today's live will be the last day lah of the live of the chapter 17 series of inheritance and genetic control. So, for today's topic, we will proceed to regulation or gene expression. Actually, is chapter 17. The seventh topic lah. The last two topic of this chapter, regulation of gene expression. Okay, so for today's topic, we will see to regulation gene expression. So you have to know what is opposite system, and the opposite system that we learn is lab operon. Okay, so firstly, you have to know what is mean by operon system. So operon system actually it is the functional unit that consists of promoter, operator, and structural genes that function as transcribed as a single mRNA. So actually it is the DNA sequence. Dia adalah salah satu DNA sequence that contain promoter, they contain operator, and also the genes that can be transcribed. The gene ini this is promoter, this is operator, and the genes that can be transcribed to mRNA. The gene that can be transcribed to mRNA. So this is the definition of the operon system. So this is the gene sequence that contain promoter, operator, and the structural gene. This gene will be transcribed to form mRNA. So what is mean by promoter and what is mean by operator? Okay. So actually, the operon system that we learn is lab operon. So lab operon look like this. Dia macam ni. So this one actually is a DNA sequence lah. DNA sequence. And for this topic, we only learn prokaryote operon system. We don't learn about the eukaryote one because eukaryote is more complex to compare to prokaryote. So we learn prokaryote, and one of the example of operon system in prokaryote is lab operon system. Okay, this is the lab operon structure. So I already say the definition of the operon system actually is from here until here. It contains promoter, operator, and also structural gene. So actually this one is the gene. But it is the gene one. Structural genes that later will be transcribed into form mRNA. But since it is the lab operon contains three genes that will be involved in operon system that later will be transcribed to form mRNA. So contain lab A gene, lab Y gene, and lab Z gene. And this gene is encoded to these three types of enzyme called transacetylase, permease, and beta-galactosidase. Actually, it's beta-galactosidase. In examination, you have to write the beta in like this. Tak boleh, you cannot write in alphabet form. Like in word form, tak boleh. You have to write in this Latin form. In Latin form one. Okay. But if you see that actually it contains regulator. So actually regulator is not the definition involved in the operon system. So if you write the definition of the operon system, you cannot including regulator. Regulator tak terlibat dalam operon system punya. But why we also need regulator? Because if you see that the promoter is the binding site of RNA polymerase. Sorry. Not RNA, RNA polymerase, right? You already learned transcription, right? So transcription, if we want to trigger initiation, we need promoter. So promoter is the binding site of RNA polymerase. So operator is the binding site of repressor protein. So what is repressor protein? Later I will explain. But repressor protein macam mana dia produce hasil? Dari sini lah. Repressor is produced from regulator. R is regulator. So that is why we have to mention also regulator. But in operon definition, kami tak include regulator because in this diagram kami include regulator because of the operator require repressor protein. So reproducible protein is produced for regulator. That is why we kami tulis regulator lah. So that is why kamu tengok ada satu one column at here. Ada satu column at here, right? Because actually the distance between regulator and promoter is very far. If you see that promoter, operator, and structural gene, they are close together. Normally are close together. But for the distance between regulator and promoter, it is downstream very far lah. Downstream for promoter, not downstream, upstream for promoter very far away lah. Ada banyak ada dalam tengah-tengah ni ada banyak DNA sequence lagi. So that is why regulator is not involved in the opposite system one. Okay, so this is the definition. So promoter is the site for attachment or binding site for RNA polymerase. Operator is the binding site of repressor protein, and it acts as switch to either activate or deactivate the opposite system. Because you have to know this topic is about regulation. So what is mean by regulation? Means by it may be ada if you see terms regulation, either it is on or off. Either dia buka atau tutup. Either dia activate or unactivate. So for this one is activate or deactivate. Activate. Activate. Okay. Next is about strategy. Strategy is a genetic that contain genetic information to synthesis polypeptide. That is for like it is synthesis the enzyme. Regulator and then that regulator is the synthesis of repressor protein. And then what is repressor protein? So repressor protein is prevent the transcription of the structural gene to occur. Kalau ada repressor protein and then they bind to the operator, it will prevent transcription process. Maksudnya kalau ada repressor protein dekat sini, maksudnya transcription tak akan berlaku lah. Kalau ada repressor protein. If no repressor protein, then transcription will be run as normal. So this is the repressor protein function. It is to prevent transcription to occur. And then there were another two terms is for inducer and co-pressor. So inducer is the specific small molecule that bind to the repressor protein to inactivate the repressor protein by switching on the opposite system. Okay, so inducer, so actually if you see that is the presence of repressor protein. So if kalau ada repressor protein, memang dia tak akan ada transcription lah. But repressor protein is already produced because we got regulator. So how to prevent the repressor protein bind to the operator so that the transcription can be run as normal. So there we will got one molecule called inducer. Ada satu inducer, it will bind to the repressor protein so that the repressor protein cannot bind to the operator, and then the transcription will run as normal. And another one is co-pressor. So co-pressor also bind to the repressor protein, but this protein is to activate the repressor protein by switching off the opposite. So actually this one you don't need to learn lah. This one is another opponent called tryptophan. Tryptophan opponent. This one is not in your syllabus, but in your Oxford book, maybe I think Oxford book ada mention sikit. But if you refer to the NPM syllabus, actually you don't need to learn. So ini compressor awak tak perlu risau, tak perlu belajar. Tak perlu belajar. You just need to learn inducer only. Compressor you don't need to learn. Okay. Okay. Next is about the mechanism of the lag opponent. Kalau tadi awal sudah faham sebuah definisi kan. So sekarang kami nak learn the mechanism of the lag opponent. So why that is called lag opponent? Because it is depends on this gene expression is depend on lactose. Either lactose itu absent or lactose itu present. Kalau either dia lactose tu tidak ada lactose dalam sel, ataupun lactose itu ada dalam sel. So this is for the E. coli one. for the general opponent system for E. coli. Why this is called lactopol? Because it is depend on lactose. Kalau mereka ambil dari LAC ni lah. So they call lactopol. Okay. So if you see that if you see this diagram, actually this one is regulator lah. Ini regulator. Lepas tu yang ni adalah promoter. This one is operator, and then this one is the structural gene. Actually, they are also let Y and also let A. Yang ni dia tak bagi lah. Kenapa dia tak bagi? If you see this. Okay. Okay, so firstly, our regulator normally it will produce mRNA. It will be transcribed to form mRNA. And then later it transcribed to form mRNA, they will be translate the type of protein called repressor protein. Yang ni it is the protein called repressor protein. And this protein, it will bind. I already say repressor protein will bind to where? It will bind to the lab operator. Okay, it will already bind to the lab operator. So because it has the binding site, the shape of the binding shape same as the binding site of the operator. So it will bind to it, the lab operator. So after once it bind to the operator, the RNA polymerase cannot bind to the promoter already. Dia sudah tak boleh lah. Because promoter is according to here. Dia sudah tak boleh bind toward the promoter. So if you see this in the some lactose, it switch off the tak boleh bind to the operator. So this is what happen is when lactose absent. Kalau lactose itu tak ada dalam sel, repressor protein will directly bind to the operator. So this will cause the RNA polymerase cannot bind toward the promoter, and then the transcription will not produce. Karena RNA polymerase sudah tak boleh bind dekat promoter, so equally there will no any transcription process will occur in the structural gene. So our LAC-Z, LAC-Y, LAC-A gene, semua gene ni tak boleh cannot be transcribed to form mRNA lah. So that is why he say that no mRNA will be produced. Actually, it's mRNA. No mRNA will be produced. So this is the mechanism of the that occur when the lactose is absent. Kalau dia tak ada. Kalau dah senang tak ada. Repressor protein produced, it will directly bind to the lab operator, cause RNA polymerase cannot bind toward tak boleh bind toward the promoter, and then no transcription occur lah. So tak ada enzyme, tak ada mRNA, tak ada enzyme produce lah, because of lactose is absent. So this is recalled as the lab operator is switched off. Dia susah switch off lah, because no transcription occur. Inilah kami panggil switching off. Deactivate, or other terms is deactivate. Okay, next is about lactose. Kalau dia ada, if the E. coli cell contain lactose, so macam mana? So if you see that actually it is same, the regulator, this is regulator, ini promoter, ini operator, and then this is the structural gene. You will see that the regulator also will produce synthesis repressor protein. But since the lactose is present, so our lactose molecule is called allolactose. So it is act as the inducer. I already say inducer. So normally if you normally in structural question, if you call label but labeling, you can write either allolactose or inducer lah. Sama saja. Allolactose or inducer. Allolactose will bind toward the repressor protein, because there are another binding site for the allolactose. Yeah, you have to mention allolactose. Tak boleh hanya cakap lactose because it is the another modification of the lactose So actually it is allolactose. It will bind to the repressor protein. Bind toward the repressor protein, because repressor protein got another binding site for allolactose one. So after it bind to the reposer protein, it will inactivate the reposer protein. It will change the shape. The shape of the reposer protein will change. Dia sepatutnya yang macam ni. Sekarang lactose bind, dia sudah jadi bengkok lah. It already changed the shape. Shape is altered, so it will cannot bind toward the operator already. Because the shape of the binding site already, the binding site already changed. So dia sudah tak boleh fix to the binding site of the operator, so dia tak boleh bind toward the operator lah. Okay, so since no replacer protein bind to the operator, so by our RNA polymerase can bind toward the promoter. Dia sudah boleh bind, can bind toward the promoter, and then since they are RNA polymerase, so the mRNA of the structural genes A, Y, Z will be produced. So there were actually got three mRNA lah. One mRNA is for Z, one mRNA is for Y, and one mRNA is for A. So in this case, you will see that there was start codon and stop codon here. So start codon means that yang ini kawasan ini adalah untuk Z punya. Kawasan ini adalah Y. Yang kawasan ini adalah untuk A. A1. So these three respectively mRNA will produce beta-galactosidase, permease, and trans-acetylase. Will translate lah. This is for translation. But you have to use translate lah. The mRNA will be translated to form these three enzymes, and then this enzyme will be used to hydrolyze the lactose. It will be used to hydrolyze the lactose molecule. Hydrolyze lactose molecule. So this is the mechanism where lactose is present. So hanya satu perbezaan sahaja, bahawa the replacer protein cannot bind toward operator, and then this will cause the operator is switch on. Dia sudah activated, become activated. So transcription can be occur, and then got genes mRNA produced, and then so mRNA will be translated from three and respectively enzyme. So yang paling penting hanyalah replacer protein cannot bind toward operator. Why the replacer protein cannot bind toward operator? Because the presence of the allolactose. So allolactose will bind toward the replacer protein to inactivate the replacer protein. So already changed the shape. So the replacer protein cannot bind toward the operator. Tak boleh bind, RNA polymerase boleh bind to the promoter lah. RNA boleh bind toward the promoter, and then the transcription start. mRNA produced, and then the mRNA will be translated to the enzyme, and this enzyme will be used to hydrolyze the lactose. So this is the overall mechanism of the operand lah. Okay, so hopefully this topic is actually very easy topic lah, tak susah. Just it is the gene regulation only. Okay, so hopefully this live is end lah. Cepat sahaja. So hopefully you can scan this script platform so that I can know my performance. So you can leave now. It may leave now. So thanks for your guys' support and have a nice day. Thank you.